5, the last of our foundational lectures. Thursday is going to be drug discovery. I'll let you know what questions from that lecture will be on the exam. So I think I mentioned before, it's going to be a two-part. I was going to do a brief lecture on drug discovery process. Dr. Anupam Jingaran will be here from MSU Innovation to talk about intellectual property and those sorts of things outside of the actual science, but are just as important in developing in drug discovery and development these days. But nothing from his lecture will be on there. That's more background stuff, and very little from the drug discovery lecture. But to that end, a week from today is going to be exam 1. It'll be right here in this room. If you have a visa, if you're a visa holder from the RCNP, I forget the acronym, I apologize, but if you're holding a visa for the class, it will be, not a visa for being in the country, but a visa for the class. Life Sciences B448, so 4th floor of the building, Life Science proper next door, at the same time as this exam. So I'll be here or there. Review session will be this Sunday night at 7 o'clock. I'll put a Zoom link on D2L probably sometime this weekend. Feel free to email questions if you have them. Show up and ask them. I'll record it and then post it that night. And I think that's all for announcements. I will say, yes, this is our last foundational lecture. We're going to talk about neurotransmitters specifically today that are going to be important for the drugs that we talk about. But I have added a few slides at the end of the lecture that are not posted to D2L about in vivo experimentation. So doing experiments on animal models is a large way that we have been able to understand what we do understand about the drugs that we'll be talking about for this class. So understanding that kind of experimentation is very important. These few slides are just kind of a primer on things. Once we get into the actual drugs we'll be talking about, I'll talk more about the specifics there. When we get to the last slide that's published, either please don't fold everything up and get ready to bounce as soon as you can, or do it very quietly for those who would like to stay after and hear. It's only a few slides. I'm just going to talk a little bit, but it's just a bit of a setup for what we'll get into. And again, when we start talking about the drug specifically, I'll get into the specific in vivo experiments we've done, so we'll cover all of that. And this is just sort of a introduction for those who aren't familiar with that type of experimentation. All right, that's all the announcements. Any questions before we dive into Module 5? Good? All right. So, nervous system sites of drug action and toxicity. This statement in the beginning is not too profound to state, but is central to understanding everything that we'll be talking about. And we'll see this pattern throughout today's lecture, as well as the drugs we're talking about. So, presynaptic neurotransmitter release binds to receptor on postsynaptic neuron, and how that gets toggled is central to how all of the drugs that we talk about work. But neurotransmitters we'll talk about today: dopamine, norepinephrine are monoamines. Dopamine definitely the most important for discussing drugs of addiction. Serotonin, also very important for several of them. And then the rest are acetylcholine, and then glutamate and GABA, the activator and suppressor, if you will, of neural activity in the brain. We're going to go over that, and then again, a couple slides on in vivo experiments. And yeah, diving in. So, This is just a general look at what we're going to talk about for this lecture, and as you will hopefully see, there are patterns to everything that we're talking about. But I threw this one in there just as a way to organize, or if you need help with organizing what we'll be discussing, organizing this material, I feel like this is a good way to break down at least the portion on the actual neurotransmitters. So here's two nerves, two neurons, they synapse here. This is a blow up of this. Here's our presynaptic nerve. Here's our postsynaptic nerve. The major players involved here, presynaptic and postsynaptic nerve. Here we have a synaptic vesicle with loaded with these blue circles, which are our neurotransmitter. As we see, this will descend down into the terminal, bind to the membrane, release the neurotransmitter out into the synapse, where it can bind to its target receptor. Obviously, this ion channel is just a general representation of receptors. There are also the GPCRs that are very important for us a bit differently, but this is just a placeholder for a targeted receptor on the postsynaptic nerve terminal, and the effector proteins that then are activated from there. So, in the presynaptic nerve terminal, knowing which neurotransmitter we're talking about obviously is very important. The synthesis of them will be important, and I'll highlight, you don't need to know all of the synthesis for all of them, but there are a couple of enzymes I'd like you to know, and maybe some precursors that are important, and then the breakdown of them. So what we'll see is cycling happens. This isn't just released, neurotransmitter isn't just released out into the synapse. There's also transporters for bringing the neurotransmitter back up into the presynaptic nerve terminal, and then we'll see the neurotransmitters get broken down or they get recycled back into use. So this breakdown portion is really what happens after release into the synapse of that neurotransmitter. Then on the postsynaptic nerve terminal, the receptor and effectors that are important there. So those are broad brushstrokes of what's important to know about each of the systems we'll be going through, okay? Diving in, dopamine. This is the one that does all of the, well, that. This is the central player in addictive circuitry, right? This is where our reward pathway is based. Several different primary pathways in the brain are important. Mesolimbic or mesocortical pathway, usually referred to as mesolimbic, nigrostriatal pathway and tuberoinfundibular pathway. So shown here, we see in dark blue, this is our mesolimbic dopamine pathway. So we're going midbrain and top stem into the forebrain. Then mesocortical is the big one where this goes from, here's our VTA, out into nucleus accumbens here, and then into the PLC, all right? Nigrostriatal is a little bit deep to that, but goes up into midbrain areas from the substantia nigra into thalamus, striatal tissue in here, okay? And then the last, the tuberoinfundibular, is more for hormone release in that, since it is based in the pituitary, or it comes out into the pituitary and can trigger the release of hormones, et cetera, et cetera. We don't talk much about that during this class, but it is important as far as dopamine pathways go. So, that's the three pathways, and that's and where they lay out in the brain, and that's important. But a bit more specifically, synthesis of dopamine comes from breakdown of the amino acid tyrosine, right? So these are known as, so this is one of the monoamine or monoaminergic. Here's our amine here. This is a monoamine molecule. So tyrosine is the precursor molecule for dopamine. And the first step in the pathway is conversion of tyrosine to dopa, all right? Dihydroxyphenylalanine, okay? Tyrosine hydroxylase is the enzyme that facilitates that. I'd like you to know that enzyme. No tyrosine hydroxylase. We'll talk why that's important, we'll get to. Don't worry about dopa. Don't worry about dopa decarboxylase. Tyrosine, TH, as it's abbreviated, tyrosine hydroxylase, and then onwards to dopamine, is important, okay? Now looking specifically at a neuron, again, we're gonna see this pattern over and over today, and I hope it doesn't get too annoying, but tyrosine gets broken down, or eventually is converted into dopamine, packaged into synaptic vesicles, released out into the synapse where it binds to a dopamine receptor, and then will cause some intracellular change in a postsynaptic neuron, okay? Broad brush strokes. A little bit more on that, okay? Again, here is our presynaptic nerve terminal, here is our postsynaptic nerve terminal. Tyrosine broken down by tyrosine hydroxylase, converted to DOPA, which is converted to dopamine, abbreviated DA. That's our blue balls here. Packaged into a vesicle by the activity of this protein, VMAT, vesicular monoamine transporter, just no VMAT. That is what is responsible for pumping dopamine into an endosome to create a synaptic vesicle. Once it is filled, it can then bind to the membrane, releasing its cargo out into synapse, where it will target a dopamine receptor. 5 major subspecies or members of this class are recognized, D1 through D5. D1 and 5 are similar, D2, 3, 4 are similar. These are all GPCRs. We'll just talk about D1 and 2 here, representing their class. D1 is bound to G alpha S, okay? Remember, this is a GPCR, so those are coupled to G proteins. The specific protein that this is coupled to is G alpha S, just abbreviated GS here. Dopamine D2 type receptors are also GPCRs, but they're bound to G alpha I, or represented by GI here. And what they do, GS will stimulate adenylate cyclase. This will convert AMP, adenosine monophosphate, into cyclic AMP. So adenylate cyclase converts AMP to cyclic AMP. That cyclic AMP is then an effector molecule or second messenger that then triggers within the cell. GI does the opposite. I stands for inhibition. This is an inhibitor of adenylate cyclase. Excuse me. And it prohibits the formation of cyclic AMP. So S for stimulate, I for inhibit. Dopamine binding to their receptors, it binds to D1, it'll increase cyclic AMP levels, binds to D2, it'll decrease cyclic AMP levels. Then, any residual dopamine that's left out here in the synapse is brought back into the presynaptic nerve terminal by DAT, dopamine transporter, all right? This protein sits in the membrane and actually pumps the dopamine back up into the synapse, where it can either be broken down or recycled back into a new vesicle. This is broken down by MAO, the enzyme MAO. Monoamine oxidase, just need to know MAO. This enzyme will convert dopamine to DOPAC. Worry about that, just what it is. If it's not broken down by MAO, it is repackaged back into synaptic vesicle by VMAT, okay? So, important things, tyrosine is converted, first step by tyrosine hydroxylase, eventually becomes dopamine, which gets packaged into a vesicle by VMAT, that's released out into the synapse, and it can bind to its target dopamine receptor, either D1 family bound to GS or D2 family bound to GI. S will stimulate adenylate cyclase to make cyclic AMP. I will inhibit adenylate cyclase to prevent it from making cyclic AMP. Residual dopamine in the synapse is uptaken by dopamine transporter, or DAT. That is then broken down by MAO, or it is repackaged by VMAT. All right? That's the basic cycling of dopamine. Why all that's important, we'll talk about with the specific drugs, but just knowing that. Flow is important. How does that sound, everyone? Good. Straightforward. All right, ready for redundancy. Good thumbs up, like it. Norepinephrine. Locus coeruleus pathway is norepinephrine. Write that on the back of your hand so you don't forget it during an exam if you don't know that already. Locus coeruleus is norepinephrine. Primarily. I'll get in trouble for that, but that's what it is. As it shows here, we have the origination of this pathway in the rostral raphe nuclei, which makes up our locus coeruleus, starts our locus coeruleus, and then we can see projections out into cerebellum, and then out into the midbrain and the PFC, similar to dopamine, but solid. All right? Locus coeruleus, norepinephrine. LC equals NE. This will affect a sympathetic nerve system. I don't know if I should pull this or not because we really don't talk much about sympathetic, parasympathetic pathway here, but this is a major player in that pathway, not just in terms of how it works in the brain, because there is overlap with reward pathway and norepinephrine cycling. However, epinephrine and norepinephrine are major players in sympathetic nerve system, which is part of the PNS or peripheral nerve system, as shown here. So here's our CNS reaching out into these ganglia here, which then innervate all kinds of different organs and areas of the body outside of that, okay? So, epinephrine and norepinephrine synthesis, these are also monoamines, also come from tyrosine, okay? We just looked at this. Tyrosine hydroxylase. Dopamine, or we go one further, dopamine beta hydroxylase, or D-beta H, know this one, will convert dopamine to norepinephrine, all right? Within the adrenal gland, you also have phenylethylamine and methyltransferase. Don't even bother with that, but you convert norepinephrine to epinephrine, okay? Just know that norepinephrine can be converted to epinephrine. And it's all based on what the body needs at the time. But, okay, tyrosine, TH, dopamine, D-beta H, norepinephrine, and epinephrine. Those are the hot words there, what have you. So, this is a different representation of exactly what we just looked at for dopamine, but adjusted for norepinephrine, all right? We got tyrosine, we're gonna make dopamine. Dopamine gets packaged into a vesicle by VMAT. Now here's what's important. Here's our DBH within the vesicle. So dopamine is converted to norepinephrine within the vesicle. Then that norepinephrine will do exactly what we just talked about. Release into the synapse, hit specific receptors for norepinephrine. Alpha and beta here are adrenergic receptors. Not going to get much into those, but just for sake of clarity, that's what alpha and beta represent here. But back to what's important. Any residual norepinephrine that doesn't hit receptors gets brought back up by NET, norepinephrine transporter, and then the same thing happens. It can either get broken down by MAO or repackaged by VMAT. In which case you won't need conversion, right? Because you're already at norepinephrine. But bottom line is that VMAT also works on norepinephrine, as the name states, vesicular monoamine transporter. So all the monoamines are acted on, or they're substrates for this enzyme. All right. Serotonin. Primarily in our raphe nuclei, caudal and rostral, right? As we see here, our rostral and our caudal, see projections from here into cerebellar area from both, and then again from midbrain out into cerebral cortex, or to our PFC. And this one is made, serotonin is made from So the amino acid tryptophan is reacted on by TPH. While they're saying it, you don't need to know it. Just know that serotonin comes from tryptophan, all right? There's structure there. Oh, right, and don't worry about structures. You're not gonna be, I won't show you a picture and what is this molecule or anything like that. Again, this is more theoretical. You don't need to do math. You don't need to do structures for this class. But you gotta know your theory. Serotonin. So, similar orientation as we just talked about. Presynaptic terminal, postsynaptic terminal. Here's our synapse. Tryptophan gets created into 5-HT, 5-hydroxy tryptophan, or tryptamine. 5-HT is abbreviation or representation of serotonin, but this also gets reacted upon by VMAT to form the vesicle. This neurotransmitter can be released out into the synapse where it can bind to serotonin receptors. 5-HT1 and 5, so these are also GPCRs. 1 and 5 are coupled to GI, 5-HT4, 6, 7 coupled to GS, right? So same thing happens there. You bind to the GS coupled receptor, you turn on adenylate cyclase. Bind to GI coupled receptor, you turn off adenylate cyclase, okay? And then here, residual serotonin in the synapse is brought up, is reuptaken by serotonin transporter or SERT, and it can be broken down by MAO or VMAT, all right? So, very similar pattern. Just need to know a few of the specifics for these. Okay. On to cholinergic pathways. As shown here, pedunculopontine nucleus, lateral dorsal tegmental nucleus are cholinergic pathways. So pathways, again, in similar areas we just talked about, we're going from midbrain out into cerebellum, and then midbrain out into PFC area. Here, a couple different receptors we'll talk about in just a minute, but this one, acetylcholine, is assembled from acetyl-CoA and choline, all right? So this is a unique one in that we have two molecules that are assembled to make this, to make a molecule of acetylcholine, all right? All of the precursors and whatnot are not important, but know that acetyl-CoA and choline are assembled to make acetylcholine. That gets released out into the synapse where it can bind to acetylcholine receptors. These are either nicotinic receptors or muscarinic receptors. Nicotinic receptors are ligand-gated ion channels. Muscarinic receptors are G-protein coupled receptors. The other unique thing about this is acetylcholine is actually broken down in the synapse. There are acetylcholine esterase enzymes in the synapse that break this down, freeing up choline and acetate. Acetate will just get put back into the pool of building blocks for everything in our body. But choline is then released, and then it can get re-imported back into the presynaptic nerve terminal for assembly into acetylcholine again. Great. And this just states that, again, in a bit more simpler form. This is probably a better one for studying from. It's a little less muddy. But again, CoA, not so worried that you know what happens in the mitochondrion, but it is assembled into acetyl-CoA when you add choline to it, forms acetylcholine, released out into the synapse where it will bind to muscarinic or nicotinic type receptors. Its breakdown enzyme, acetylcholinesterase or ACHE, will break this down into acetate choline, which can then be recycled back up into the presynaptic nerve terminal for use in the next cycle. All right. And again, nicotinic, muscarinic, nicotinic are ligand gated, muscarinic are G protein coupled, and I hope I switched those, I did not, but quick aside everybody, I'll get back to that story. Couple of neuron types that are going to be important for discussing what we're going to talk about: interneurons and projection neurons. Interneurons are housed within one area of the brain. So here we're looking at acetylcholine that will synapse on a dopamine neuron, as well as, in the VTA as well as the nucleus accumbens. Our two acetylcholine neurons are interneurons because they're housed within the VTA and the nucleus accumbens respectively. Dopamine neurons are projection neurons. They go from one area into another. They project into another area. So here, dopamine, the soma would be in the VTA. Its axon will actually synapse in the nucleus accumbens, and then this dopamine neuron, soma here in the nucleus accumbens, and then projects out into the cerebral cortex. So remember that. Interneurons are local, projection neurons go long distance, but those are important to remember for this course, for understanding what we'll be talking about. I want to switch these orders up, and I apologize, but back to acetylcholine, just some examples of what I just talked about. In terms of nicotinic and muscarinic receptors. So this is an example of a somatic motor nerve here that starts in the spinal cord and projects out into the periphery and synapses on a skeletal muscle tissue. This will be for an excitatory transmitter that will cause the muscle to flex. And then an autonomic motor neuron here, so essentially the same area, but it's going to more, instead of a cardiac, or excuse me, a skeletal muscle, this will synapse on a smooth or cardiac type of muscle. These can both be, or the neurotransmitter here is acetylcholine, as this diagram shows. Norepinephrine can be one depending on where specifically we're talking about, but I'm talking using this example to demonstrate the difference between a nicotinic and a muscarinic receptor, because nicotinic acetylcholine receptors with the skeletal muscle and muscarinics with the smooth or cardiac muscle. Now, this plant, of course the name escapes me, but you can isolate curare from this. This works as to block block nicotinic receptors and cause paralysis of skeletal muscle. This is one of those paralysis agents that are used for hunting. A lot of indigenous tribes have used this or isolated this and allow it to hunt certain prey because they can nail them with a dart that's dipped in curare and freeze their prey, and that's how you get them stop running away by hitting it with this. And then something we'll get into a little bit, but something that's getting a lot of attention lately, fly agaric mushrooms, or Amanita muscaria mushrooms, these very distinct red capped with white dot mushrooms, can act as activators at muscarinic receptors, causing rapid activation of smooth or cardiac muscle. And all of the fun things that happen there we'll get into, but just a couple examples of the difference between nicotinic and muscarinics in these systems. Okay, back to our neurotransmitters. Glutamate comes from glutamine, so the precursor for that is the amino acid glutamine. Glutaminase is the enzyme that causes this to convert to glutamate. So used as an amino acid or a neurotransmitter, but the same thing happens here. It gets packaged into a vesicle by V-glutes. Those vesicles will release glutamate out into the synapse where it can bind to its target receptor, NMDA, AMPA, or glutamate receptor of the non-ionic ion channel type. Any residual glutamate is then taken up by glutamate amino acid transporter, EAAT. Don't worry about this side of the equation. We don't go into that much detail or this nuance in it. Just know glutamine converted to glutamate that's packaged by V-glutes, hits glutamate receptors, transported by EAAT or glutamate amino acid transporter, brought back up into the presynaptic nerve terminal. Yeah, this just shows the breakdown in a neighboring cell, but don't worry about breakdown of glutamine and that. Just glutamate, V-glute, glutamate receptors we'll get into, and then EAAT for transport back up into the presynaptic terminal. Lastly, GABA, very similar to glutamate. As this then gets, glutamate can be converted to GABA, GABA aminobutyric acid, there's no GABA, and then same thing, release out into synapse. GABA A receptors are ion channels, GABA B or GPCRs, GABA transporter reuptakes any residual stuff, and then all this stuff happens, but don't worry about that. All right. That's our neurotransmitter list. Questions, please. A lot of similar pattern again. Hopefully nothing too surprising, hopefully nothing new, but you know, that's the extent we really need to know for this class. So, I'll dive right into the in vivo stuff. A lot of the experiments that we'll be talking about again come from experimentation on animals. One of the most basic observation chambers looks like this if you're not familiar with them. Operant conditioning chamber is the most proper title for this. This is what they actually look like. Well, they come in lots of different shapes and sizes, but this is the most basic one. The electrified grid and the loudspeakers, I just left that on there because I found this diagram. I thought it was hilarious because that's, yeah, I don't remember when they used to do research with the electrified grid on the bottom. I'm sure it still does happen, but really look down on that kind of thing lately. What I bring this up for is the main parts of this chamber that are important are like this. Well, back up. So we have a chamber, and we put a mouse or a rat or a rabbit or something or a person in this chamber and observe them. And the things that are really important here will be here the lights, here the response lever, and here the food dispenser. These individual details are not as important as the pattern that is needed here. So, yeah, it can be lights, but this is more stimulus. Okay? Stimulus, response, reward. Those are the three things that are really important in these chambers, especially when you are examining illicit drug use. Stimulation can be light, it can be a sound, it can be a picture, it can even be a dose of a drug, okay? But just remember, stimulation, and then there's supposed to be a response. Here, it's a lever press. A lot of times it's a lever press, sometimes it's a nose poke. They have a little hole that an animal can stick their nose in. But it's something that shows you're responding to stimulus, okay? And then you have a reward. A lot of times it's this food dispenser. Here, you know, they want to see how many times this rat is going to press the lever to whenever the green light comes on. Green light comes on, press the lever, you get a food pellet as a reward. Quickly will establish in that rat's brain that if the green light comes on, if I touch this thing, I'm going to get food. Same thing happens with drugs. A light comes on, I touch this lever, and a lot of times, and we'll see this specifically, there's actually cannula placed, like a microdialysis tube is placed right in, surgically embedded in the rat's brain that will deliver drugs to the brain. And yeah, then in those cases, you don't usually have the food dispenser because that's its own thing. It's more observing the pattern of behavior that happens after you get that dose, but please just remember that. In an observation chamber, you have something that provides a stimulus, something that records a response to that stimulus, and then something that'll either be the reward or some recording of that response to the stimulus, all right? Those are the real three things that are important to these. Okay, last couple of things, sort of random other types of in vivo experiments that are important. The elevated plus maze is a way of examining anxiety in, well, rodents specifically. If you're not familiar, it looks like this. It is a platform that is shaped like a plus, right? It's just a platform with these two forearms or two branches, however you want to say it, but looks like a plus. Two of these arms are covered, okay? And the idea is the open arms, if you're traveling on this as some rodent, you can actually look down and see how high up you are. And rodents have a fear of heights similar to a lot of mammals. So they don't want to go there if it triggers anxiety for them. They'll spend a lot more time in the closed arms. But this is how you can measure anxiety. If you are developing a drug or working with a drug that decreases anxiety, like benzodiazepines or alcohol, you can test in this situation to see how anxiety decreases with doses of your anxiety decreasing medication. And what you'll see here is, well, here's some actual data on the bottom. So they actually will track how much this animal will cruise around and explore, because curiosity and exploring is a natural phenomenon within mammals. And you throw a mouse into a cage, after an acclimation period, it's going to cruise around and just explore that cage because it's curious of what's going to happen. Same thing in this type of, same behavior in this type of experiment. However, as we see down here, there are two very distinct patterns of traveling around, all right? So on the top, this would be an example of a mouse that is not undergoing anxiety or is experiencing calmness, if you will. Because as you see, there's a lot of wandering on all four arms, or in the open and the closed arm. Excuse me, man, stop still. And the bottom is an looks like anxious behavior, right? So we can assume that these are the open arms here because this looks like there's a lot more time spent and distance traveled out into the safe closed arm with very little exploration out into the open arms, right? They didn't go all the way out there because they get out here and that's enough and they come back. So this is a type of data pattern for traveling when you're anxious. This is calm or non-anxious behavior, all right? Elevated plus maze. Last one, water maze. This is for memory. So if you are investigating drugs that can affect your memory, I can't remember which those are right now, but you could do that in a water maze. So this is really just a pool of water that has well, water in it, and usually chalk or something to make it opaque. So if you're swimming in the water, you can't see what's going on, but it's shallow water, but it'll force a mouse usually to swim. And then you have a platform in there, all right? And the idea is you drop the mouse in there and you allow it to swim with the thought that it will eventually find the platform, because mice don't like to swim. But then you record how it moves around in this maze or in this water, and you can see different patterns here. And there's different types of hunting and searching and things, the different, yeah, you can check out this paper if you like, but the different dashes and colors and whatnot are different specific types of behavior. But I bring this up because as a mouse will build memory in this situation, first time it's exposed to a chamber would look like this. It's going to cruise all over the place. It doesn't even know that there's a platform there. It has to learn first that there's a platform there and then learn where it is. So these types of patterns are what you'll see. Once it's been exposed to this a number of times, it will look more like this pattern here, where it's specifically looking for something, and they usually end up, you know, the end of the experiment will be when it reaches the platform and gets up and it's safe and can rest for a minute. So, yeah. But those are types of experiments that we'll be talking about. I think that's it. Yeah. So, practice exam is posted and open. Please take that if you would like. Read from today's exam 1, Thursday's drug development. Yeah, hope to see everybody Thursday.